This is another one of the short talks chosen from the poster abstracts. It's one of our local collaborators here from Lawrence Livermore National Lab. Erin Nuccio is a postdoc in Jennifer Petridge's lab and came from nearby UC Berkeley in Mary Firestone's lab. And she, I think in our first talk today, we saw some of the power of these isotopic analyses that Martin showed. And Erin will be revisiting this using isotopes to track the pathways that carbon take through decomposition in the soil. Thanks.
Okay, so thanks for the opportunity today to tell you about some work that has led to 2 projects at the JGI and EMSL that we just started this year. So the work I'm going to tell you about is an ongoing multi-institution collaborative effort to understand how microbes transform and ultimately stabilize root carbon in soil. So the image I'm showing you here is a picture of roots growing through soil. So soil is a large dynamic pool of carbon. It's the largest terrestrial sink of carbon, and it has more carbon than the atmosphere and ocean surface ocean combined. And roots are the primary input of carbon into soil, so it's important to understand how plant microbial interactions influence below-ground carbon storage. Now I'd like to draw your attention to this plant-soil interface. Can you see the cursor? Great. Which is called the rhizosphere. This is a critical microhabitat in soil, so it's quite small, about 1 to 2 millimeters in diameter, but it provides a disproportionately large input of carbon to the soil, so it can account for about 30 to 40% of soil organic carbon input. And this zone performs many important functions in soil, including enhanced decomposition rates, as well as repressed decomposition rates in the zone, as well as increased nitrogen turnover rates. So we've observed higher chitinase and protease rates, as well as nitrogen mineralization rates in this region. So this is a conceptual map that illustrates for our project the movement of carbon from fixation to stabilization in soil. So carbon is fixed by the plant, and then this is then introduced to the soil as rhizodeposits and root detritus. This is then consumed by the microbial community, which incorporates this carbon into their bodies. When these cells die, this material can become sorbed to mineral particles in soil, and this can physically protect the carbon from being taken up again and act as a carbon sink. And then, however, this material can be re-liberated and reintroduced back into the microbial community. So for my talk, I'm going to focus on the first half of this cycle, which we colloquially call the cradle. So this is the movement of rhizodeposit carbon and root detritus carbon into the microbial community. And so our basic approach is to combine stable isotopes with microbial characterization to help us tease apart food webs and track how microbes process carbon in soil. And so what I'm showing you here is a 16-chamber facility that we've built at UC Berkeley, which we've been using to simulate elevated and ambient CO2 conditions, as well as to introduce isotopic tracers like 13CO2 for pulse labeling and continuous labeling of plant material. And this person standing right here is Shenjing Shi, and she headed the first project I'm going to tell you about. And so this project is trying to determine how elevated CO2 alters the succession of rhizosphere microbial communities in soil. And so the basic approach is we have Avena fatua, which is a grass that's common to Mediterranean grasslands throughout the world, growing in microcosms under ambient and elevated CO2 conditions. And first, we're going to look at how roots change the soil bacterial community over time. So this is an ordination of 16S Illumina data, and this blue circle represents the bulk soil. That's soil that's about 4 millimeters away from a root. And then this green circle is going to represent the rhizosphere community. So at time 0, we only have bulk soil because we have no plant growing. And so after 3 weeks, this community diverges from the initial starting population. And as the plant matures at 6 weeks, senesces by 9 weeks, and then dies by 12 weeks, the community undergoes a succession that continually progresses away from the initial starting community. If we look into deeper into what's going on here in terms of the community composition, this is a phylogenetic tree that's showing the response to the dynamic taxa over time. So this inner ring is the earliest week, week 3, and the outer ring is week 12, the latest week. And taxa that are enriched are in blue, and then depleted are in red. And so in general, communities that are enriched or depleted in the rhizosphere are phylogenetically clustered. And so this is, you can see the alpha proteobacteria and beta proteobacteria in general were enriched in the rhizosphere, as well as Bacteroidetes, while the acidobacteria and actinobacteria tended to be depleted in the rhizosphere with some exceptions. And so the acidobacteria and beta proteobacteria had the earliest response in the third week. And I'm going to focus in on the beta proteobacteria because this actually has some interesting things going on in terms of the assembly of these communities. So you can see that there's a block here of taxa that were enriched in the rhizosphere that started in week 3 and were enriched throughout the entire life of the root. And then there's a separate clade here that started out enriched but became depleted over time. And then finally a third population that didn't really respond to the root initially, but then eventually over time became depleted in the rhizosphere. So this is showing different lineages of bacteria that are showing different assembly patterns in the rhizosphere. Now, if we look at the fungal communities, these show actually a succession as well, but it's different from the bacteria in that it's delayed. So here's our bulk starting community, and that at 3 weeks and 6 weeks, the community really hasn't diverged much from the original starting community. But when the plant senesces and dies, this community really becomes much different from the initial starting community. So if we look at how elevated CO2 influence these communities, in a single growing season, we didn't see much influence of elevated CO2 on the composition of these communities. However, by geochip analysis, we did see an influence of elevated CO2 on the functional genes in the rhizosphere samples, but not in the bulk soil samples. So this seems to make sense since the rhizosphere is really where the high input of carbon is coming into the soil. So now I'm going to move on to looking at the influence of root exudates on litter decomposers. So this has a similar setup to the first experiment, except now in addition we have to having a 13 CO2 label in the atmosphere of these chambers, we've incorporated root litter that has a 15N isotopic label incorporated into the chopped up root material. And so the goal of this study is trying to determine the microbial and microfunnel populations involved in litter decompositions and try to determine functional guilds in these complex soil communities. So for example, questions we're trying to ask are which organisms prefer root litter versus root exudates? Are there communities that consume both substrates, or do we see evidence for specialists in these communities? And so the way that this analysis works, or the approach we took, was that we sequenced the total soil RNA using RNA-seq, and then we shotgun reconstructed these 16S using eMERGE. And so this allows us to do a PCR-independent analysis of the bacterial, archaeal, and eukaryote populations. And then we designed probes for a microarray to investigate the ecophysiology of these organisms using a method called ChIP-SIP. And so basically how this works is we use a machine called a NanoSIMS, which is an imaging mass spectrometer. This generates an isotope map of surfaces, in this case a microarray surface. And so this allows the visualization of RNA that's been hybridized to these probes that's incorporated an isotopic label like 15N or 13C that was originally derived from the substrate. So to first look at the communities that responded to the litter and the root. So this is another ordination of bacteria and archaea. BL stands for bulk litter, RL stands for rhizosphere litter, R is rhizosphere bulk. And so you can see that the bulk litter soil had the most distinct community. All these other communities were also distinct for the bacteria and archaea, but the bulk litter was by far the most distinct. And in the eukaryotic populations, we also see that this bulk soil containing litter had a distinct community, and that the eukaryotic communities otherwise were more variable and not as distinct by habitat as the bacteria and archaeal communities. And so I'm going to focus in on these litter-containing communities to show you some basic rRNA abundance. And so in the bacterial and archaeal populations, a large amount of the rRNA is actinobacteria, which we don't see in the bulk litter populations. Whereas in the bulk litter, the Bacteroidetes and Firmicutes had a greater amount of rRNA. And then interestingly, we can look at the eukaryotic populations. So in the root plus litter community, it's about a 50-50 breakdown of fungi to protist rRNA that we observed. And then in the bulk litter communities, about 70% of the rRNA we detected is protist rRNA. And so this includes a large number of amoeba. And so this introduces the possibility that there could be some amoebal grazing of some of these bacterial communities. And so there's a grazing effect that is possible that would potentially be undetected if we hadn't done a multi-domain analysis. And so finally, trying to determine some guilds within these communities. So this is a chip-SIP analysis of who ate what. This graph shows phyla on this axis, and each of these dots represents an OTU. And if you fall on this side of the graph, you have more 15N, so you preferred to eat litter. And if you fall on this side of the graph, you preferred to eat exudates, so you're more enriched in 13C. And so we have some preliminary evidence for substrate preference. So some taxa that were more to the left on the graph are the acinobacteria and the actinobacteria. The actinobacteria are kind of interesting because I told you before that the rhizosphere litter communities had a large amount of rRNA that was actinobacterial. However, we see that a lot of the carbon that they're taking up is from litter material, so that's potentially not what we would have guessed. So that's an interesting observation. And in terms of who preferred exudates, the rhizobia and ascomycota tended to prefer rhizodeposits. And so this is a basic overview of where we're planning to go with this project. So we were just funded for a JGI MSL project this past year. And so our basic goal is to try to put genes on the arrows in this diagram using transcriptomics and proteomics of the rhizosphere and the surrounding soil. And in order to assist that work, we're going to couple these analyses with isotopic analyses to try to pull out some of the guilds in soil as where the carbon is going and if it's being stabilized in soil. And to assist these analyses, we're planning to expand our genomic reference library using single-cell genomics. So what I'm showing you here, the blue dots on this rank abundance curve depicts taxa that we've identified with our current transcriptomes. And then the red dots are isolates that JGI has sequenced genomes for us. And so we would like to get fill in the gaps and try to get some of these genomes that are both abundant and rare in these communities. And so this would especially help us out with proteomics analysis, which is dependent on the quality of our reference library. And so this is a genome tree of our isolates, and the size of these dots represents current proteins that we're currently able to map to these isolates. So we'd like to be able to improve this analysis. So with that, I'd like to thank the people and funding sources who made this possible, especially Jennifer Petridge and the Isotope Signatures Group, the Firestone Lab, in particular Shenjing Shi, who worked on the first project, and Don Herman, who built the chambers. Owen's group has worked on the isolates and metagenomes, as well as the folks at JGI and PNNL, who we look forward to working with in the future. So thank you.
All right, thanks. That was great. Do we have any questions? All right.